Welcome back to Lecture 2. So now we're going to be talking about the venules. We had just finished talking about capillaries and capillary beds. So now we're going to talk about venules. Venules are what pick up blood from the capillaries, or if the capillary beds are closed, then from the arterioles through that through-fare channel. Venules are the smallest of our veins, and they merge together to form veins, the small and medium-sized veins. And those are going to merge together to form the large veins. Small and medium-sized veins are the veins associated with your muscular arteries, and they tend to have the same name as the muscular arteries. So if we have the example I used before, the brachial artery, we have a brachial vein as well. We have deep brachial veins and superficial brachial veins, same with deep brachial arteries and superficial brachial arteries. Then the largest of our veins travel with the elastic arteries, or the conducting arteries. These veins don't always have the same name. So an excellent example, we have this large aorta that brings blood to all the regions of our body, and it branches off and forms a carotid artery and subclavian arteries, and branches off to form the iliac arteries. We don't have an aorta vein. What we have instead is a superior and inferior vena cava. The carotid arteries that bring blood up to our brain and to our face and our head run with a jugular vein that drains the face, the head, and the brain. So they don't always have the same exact names. These large veins have a lot of valves. The valves are important in movement of material because the veins don't have muscles, so what they use is skeletal muscles, respiratory system, to help move the material, the blood, back to the heart. And those valves keep blood from flowing from back down to the bottom. Makes sense? So if blood is flowing up from the legs, the valves will open, and then when we relax, the blood will start to pull back down, but the valves close, so blood can't move all the way back down to your foot or wherever, okay? So the basic pathway of blood flow then, I'm going to move, there we go, so I'm not in the way of the words. The basic pathway of blood flow then starts in the heart. We go from right atrium through the bicuspid valve, let me say it again. Right atrium through tricuspid valve to the right ventricle, out the pulmonary semilunar valve to the pulmonary trunk, to the right and left pulmonary arteries, to the lungs where oxygen is taken in, waste carbon dioxide is released. Blood flows back through these pulmonary veins into the left atrium, through the bicuspid or mitral valve into the left ventricle, where it is then pumped out through the aortic semilunar valve, out the aorta, and to the body. And so we're going to have gas exchange occur at all of our body tissues in the capillaries, and then blood flows back through the veins of the body to the right atrium. That's your normal pathway of blood. Alternatively, though, we can have anastomoses that bring blood in through different arteries or through different veins to a capillary bed. So here we have these different areas or ways that we can get blood to this capillary bed. And what this does is, if there's a blockage in one of these arteries, it doesn't stop the flow of blood to the capillaries, so we're still getting nutrients to those capillaries. And here it's not stopping the flow of blood back to the heart because it can still get through. If there's a blockage here, you can just flow the blood through this way and get out. Does that make sense? And if you're not sure, ask. because that's basically the function of an anastomosis, is just to give an alternate pathway so that you keep having blood flowing the proper way. Here's a shunt, like a thoroughfare channel. There's the capillary bed. Here's that thoroughfare channel that just allows us to have blood flowing directly to the veins. Whoa! And then we have this last one. I'm sorry about that. This last one is kind of interesting. This is called a portal system. And a portal system takes blood from the arteries through a capillary bed where gas exchange occurs. So the tissues become oxygen rich, and the blood becomes carbon dioxide rich. So it's going to be oxygen poor. But then instead of moving directly to the veins and going back to the heart, it goes through another capillary system. So that's through this portal vein. The portal vein will take blood that typically has a lot of nutrients in it but is oxygen poor to another region where the nutrients can be released, and then the oxygen poor blood can go back to the heart. So the best example is the hepatic portal system. Our liver takes in all the nutrients from our intestines. And so blood flows to the intestines and oxygen is released to the tissues, but the nutrients from the intestines that we have eaten move into the blood. So we have this nutrient-rich, oxygen-poor blood that moves into this portal vein. And then it's going to move to a second capillary bed in the liver where it can drop off all of the nutrients and then flow back to the heart. So that's a portal system. There are a lot of ways that we can exchange nutrients, and you've learned all of these back when we talked about movement of materials across the plasma membrane in Chapter 4, and probably if you took a general biology class here, you learned about these, or if you took general biology in high school, you learned about these. Diffusion is the movement of molecules from higher to lower concentration down a gradient. There are different types of diffusion that we talk about. We talk about simple, going straight through the plasma membrane, and then facilitative, where it goes through a protein carrier because it can't go directly through a plasma membrane. Vesicular transport is the movement of molecules via exocytosis out of the cell or endocytosis into the cell. And then we have bulk flow, where we're moving molecules along with the flow of fluids. And so molecules are either going to move in bulk flow out of a cell rapidly, that's called filtration, or back into a cell, that's called reabsorption. Or I should outside of one area is filtration, back in would be reabsorption. So there are factors that affect transport. The hydrostatic pressure is the pressure of fluids pushing material out of the cell. So the more pressure we have, all right, as I was saying, the more pressure we have pushing fluids out, the more likely materials will leave the body or leave the cells, leave the blood, and go into the interstitial regions. Colloid osmotic pressure is the pull of fluids back into the blood. Sorry. So I just said that colloid osmotic pressure is the pull of fluids back into the blood, and the pull of fluid occurs because when fluid leaves the blood, then we have a lot more proteins and other solutes in our blood plasma that can pull excess fluid back into the blood so that we don't lose all of our water, or we don't end up having a lot of pooling of fluids in interstitial regions that would cause edema to occur. The net filtration is the difference between the hydrostatic pressure and the colloid osmotic pressure then. We'll talk a lot more about this in a few more slides, okay? So let's look at diffusion. What type of molecules can move through diffusion? Oxygen small nonpolar hormones, nutrients. So these hormones would be things like estrogens or testosterones that can move from higher to lower concentration directly down the concentration right into the cell. Waste products, or into the blood, I should say. Waste products like carbon dioxide or other wastes can move from the blood or from the cells back into the blood. So these typically move from our blood to our tissues where they can then do their work, and the waste moves back into our blood and goes out of our body. Either it's going to be delivered to the kidneys or to the lungs, where we're going to expel the waste product. I just mentioned this, but here's another slide that talks about the size of molecules that can be diffused. It would depend on the type of capillaries. So small solutes can move directly through those complete capillaries through the tiny little holes called intracellular clefts. If you have larger molecules, either large proteins or just larger peptides, then they can pass through either the fenestrations in the fenestrated capillaries or the large gaps in the sinusoids. Vesicular transport is the movement of molecules from either the extracellular region inside the cell by producing a vesicle from the plasma membrane. The plasma membrane actually engulfs material and pulls it in. Or a vesicle is formed inside the cell and it attaches to the plasma membrane, and then it opens up and releases its substance into the extracellular region. And then bulk flow is the movement of a large amount of fluids with dissolved solutes in there. And they move down a pressure gradient. So you're going to move towards a lower pressure. So fluids are going to move out or into the blood, depending. And the movement direction is going to be associated with both your hydrostatic pressure, the pressure of the fluids, and the colloid osmotic pressure, the pressure of the solutes and proteins. Filtration is movement of fluid out of the blood and into the interstitial regions, into the cells. Reabsorption is the movement back into the blood. So typically on the arterial end, we're going to be filtering a lot of materials out. On the venous end, we bring them back in. And the two basic pressures that we look at are hydrostatic pressure and colloid osmotic pressure. Your blood and your interstitial regions can have a certain amount of hydrostatic pressure. The blood hydrostatic pressure is going to always be higher than interstitial hydrostatic pressure, so you're going to have more push for the fluid to leave the blood. Colloid osmotic pressure is the pull of fluid back into the tissue. So blood colloid osmotic pressure is going to be associated with the amount of proteins and different solutes and ions that are in the blood. Interstitial colloid osmotic pressure is going to be associated with whatever proteins or solutes are in the interstitial regions. Typically, blood colloid osmotic pressure is going to be much higher than interstitial colloid osmotic pressure. So here's a picture of what that looks like. Here we have here's the arterial. So on the arterial end, we have more fluids leaving. Here we have about 35 millimeters of mercury of hydrostatic pressure pushing fluids out. We have about 21 millimeters of mercury pulling fluids back in. What that means is we have approximately 14 millimeters of mercury between the two, which means fluid moves out. And over here, we have 16 millimeters of mercury pushing fluid out for the hydrostatic pressure on the venous end. And we have 21 millimeters of mercury pulling fluids back in. So when you take 16 and 21, you end up with